Lego.
You started?
Yes, it started.
Oh, okay. Hello, Miss Gaston.
And the 5 other people who watch these videos.
Yeah.
Talk louder this time.
Sorry. Anyway, it's us literally again, and we will be going over chapter 14, which is Mendel and the gene idea. So I will start. A little background on Mendel is what we're going to be starting with. Basically, he was the person who figured out the laws of inheritance with a quantitative approach. This was done by experimenting on various different plants, in particular, what's usually associated with him are peas. By doing crossbreeding and such, he was able to figure out what different characteristics he could produce from crossbreeding of several different plants. And yes, Jose will take it from here.
So the way he did it, he used the peas and he crossbred them with each other, but it was 2 different types of peas. One would, for example, be purple and the other would be white. And the true breeding, which is like the original plants that have like the for example, a trait for like the purple flower, that was like a full dominant purple flower, and he'll crossbreed it with like a white flower, which was like a full white flower, and that'd be called the true breeding parents, which is the pea generation, and their offspring, or like the first generation would be F1 generation, and that would really just show what the most like dominant trait would be. And he did this to a lot of his plants, and he kept on crossing to see like what type of traits they would get. And for the 3rd generation, instead of getting all purple ones like for the F1 generation, he had some white ones, which showed that some traits still pass on.
Like recessive traits.
Recessive traits, yes.
So they can not be present, but like show up later.
Yes.
And also, like, adding on to what Jose said, it's like, it shows that, like, because they're both, like, still there, it's not diluted by the crossbreeding or anything like that. It's still very much in the genome for the other flowers. It's like how with the peas, how they were like the smooth round ones, and then there was like the rough ones with wrinkled seeds that had the constricted pods, while there's also the wrinkled peas with the smooth pods, which goes into kind of like the alleles that are a part of that. So you have like your dominant and your recessive trait, which are part of your alleles. Alleles can be anything from eye color to hair color to if you are double jointed or not. These are all different things that can be carried on from those. So you've got the dominant alleles, which are the capitals, and then you got your recessive, which are the lowercase.
The law of segregation.
The law of segregation, yes.
Law of segregation, basically it means that not one trait is binded with another, if I'm not correct.
Yes, they separate
Yes.
during from each other during gamete formation. I cannot speak. So that's when they separate, and they end up in the different gametes. Am I saying that right?
Gametes, yeah.
Yeah, okay, cool.
Close enough.
Yeah. And so whenever you do true breeding, that's how you can that's how you test cross now. So like we had in our project with all the photos and everything, that's one of the definitions that we had. Aurelia?
Alright, so Mendel used these diagrams that we call Punnett squares. Adam's actually going to move up so he can show you just while I voice over. Here you go.
Try to speak louder because you're like in the backpack.
Okay, so as you can see on the board that Adam's holding, you have a capital P, which typically stands for your dominant allele, and then you have the lowercase p within the items as well. And so with this, you have a heterozygous like combination of this. So you could see that in the uppercase p, which is the dominant allele, and the lowercase p, making it heterozygous. And then You also have your recessive one with 2 lowercase p's, and that's your white flower. So you find the dominant allele typically tends to have offspring that are purple, and then the lowercase allele is white. And so when you cross these, which Meg is doing right now, you'll see that your ratio ends up to be 3 versus 1, where 3 of them have the dominant allele for purple, and 1 has the allele, the homozygous recessive alleles for white. I'm sorry. I can't really see the board, so I don't know what part she's on, but that is how it'll turn out.
Well, actually, it's 2 for.
No, if it's 2, it's like P and then P, and then 2.
Okay, she was trying to say if this one was capital, which it's not, but if it was, then that's what it would be. But since it's not, it's 2 purple, 2 white. But if we had a capital here and a capital here, there would be 3 for purple and 1 for white. So yeah.
Yes.
Cool.
Thank you.
So yeah, heterozygous means having 2 different alleles of a particular gene, because hetero means different, and then we have homozygous. That's just having 2 identical alleles of the particular gene.
Yeah, and like we stated earlier, with the heterozygous ones, it'll have it has to have a dominant allele and then the recessive allele, and then with the ones that are homozygous, it can either be homozygous dominant or homozygous recessive for that, and that'll determine the look of the offspring.
The phenotype.
The phenotype.
And the genotype, if you will.
And the
The
genotype,
phenotype.
which Ronnie will go more into now.
Well, a phenotype is the physical oh my god, I can't speak, manifestation of the genotype. The genotype would be like the letters in which that we refer to them as when we have ratios. It's basically, in the most basic terms, it's genetic makeup. And yes, we further our information about the genotype and / the phenotype through test crosses, which is kind of what Mendel was doing when he was using the Punnett squares, in order to figure out the initial genotype of, say, the parent generation or P generation. He would crossbreed them or test cross whatever. Yes. and yeah.
He would test cross it with a plant that he'll know is homozygous, which means they both carry the same trait. For example, if you wanted to know the color or the genotype of a certain plant, he would get a plant that is homozygous and crossbreed it and see what the offspring would be, and if the offspring would be recessive or be white, for example, be recessive, it'd show that the heterozygous,
which would be the mutant type, so you can tell that it's a recessive.
Yes, you can tell it's a recessive.
Yes, and you always do it with you always have one that you know is recessive, that you test the one that has a dominant trait with it, so you can test if it has the dominant or the recessive trait.
And then you'll be able to tell the carriers for these recessive traits, which is generically the point of these test crosses.
Yes. Should we talk about the law of independent assortment?
Take it away.
Yes. Sure, okay. So, this is when 2 or more genes are sorted, to independent parts, each pair of the alleles is, independent of each of the other pairs of alleles during the formation of the gamete. I'm still saying that wrong. So, this is only applying to, the alleles that are on, let's do, the allele pairs that are on different chromosomes. or like very far apart on the same chromosome. Like it can still be on the chromosome, just very far apart from it. And then like, because for example, the ones the peas we were talking about, they were all the different stuff that they were testing for, like the smoothness of the pod or like the wrinkledness of the peas. Those were all on different chromosomes. So that kind of like shows how the law of separation or independent assortment is, because like it varies based on the chromosome. Kind of like how like you could tie it into like these diseases we're looking at. Like with whenever you have the extra 21 chromosome, 21st chromosome.
The extra 20.
21.
Yeah. 21.
Which was like Down syndrome, so that's like affecting of like 21, but then like
the 23rd is like, oh my god, you're not with me.
The 22 sex chromosomes.
Yeah.
yes, like
And that could come with other
more
syndromes.
or more sex-linked diseases or illnesses.
For like Turner's and Klinefelters.
Yes.
Yes. All right, and so moving on to probability, this ties very closely to both of the Mendelian laws of independent assortment as well as the law of segregation, as we can compare this to like flipping a coin or drawing out cards from a deck. We have the probability ranges from 0 to 1, and we can see, and we're able to calculate this probability with the multiplication rule and the addition rule that Adam will go into later. But so with independent assortment, we can kind of compare this to tossing a coin and discarding of the last result. So if you're, how would you say this? So if every toss you have a probability of getting heads or tails, and it's 0.5, so 1/2. The outcome of this is unaffected, and you don't have to keep in mind your previous data. This will be independent assortment as you're taking both of those, and you can use the multiplication rule to find out the probability of getting a certain
Genotype or
phenotype. Yeah, and so the multiplication rule goes like this.
So yeah, the multiplication rule, you just take the probability of one thing happening and then you times it by the probability of its opposite happening. So a coin, that's 50-50, so you take half times a half and then you get a fourth. So the probability of like 2 coins both landing on heads would be like a 25% chance. And then the addition rule, it's the probability that 1 or 2 more events will occur is calculated and then added together.
So yeah, adding on to that, like it's in the like addition rule, it's like you take, because you have like the sperm and the egg, so you have all of the polar bodies from the egg. So like the possibility that the one that has the said trait that we're looking for the probability of, like it lessens by like 1/4, because only one of the eggs gets used. But whereas the sperm, it's all 4 that's produced are used, so the probability of that trait is higher than that one. So we take both from the multiplication rule into the addition rule.
Okay. To further create the distinctions for you, there are 3 general ways in which that we I would say classify different types of expression. There's complete dominance, incomplete dominance, and codominance. Complete dominance would be like the heterozygous like dominant trait. It'd be like if you had a purple flower and there was both, the genotype was like 2 capital D's, it was heterozygous dominant. That would be a complete dominance. It's completely this one color.
And the only color that comes off is one color.
Yes. And then there is the incomplete dominance, which is a common phrase for that would be like the blending of traits. So
Paint, if you will.
yes, exactly. That's a really good analogy for it. Paint, or I don't know. Some people like would use for our project. They would use like a blender. That's so you like if you had a white rose and a red rose, and you get a pink rose. That would be incomplete dominance. And then there's co-dominance. Co meaning together, so they're both of the expressions are
Being
vividly vividly present. So like if you have a cat or a dog,
a calico cat.
yeah, and they're
Particular.
like both colors are expressed. And we will show you an example.
An example of code dominance.
Of incomplete dominance
no,
coming
no,
in
no,
for
no.
this one.
No, here's incomplete dominance.
The lining makes it a bit hard to see. However, I will turn it. All right, so we are locking a pink marker right now, unfortunately. But you can see we have a red flower over here, which is typically a dominant allele that you'll find with white as the recessive allele. And so when we have this dominant allele combining with this recessive allele, we sometimes have incomplete dominance shown. And so when this red flower and this white flower creates an offspring, it may create a pink flower. So that is how we will tie
Example of
two.
codominance.
It's right here.
It's my cat.
Oh.
This is Abby. She's a calico. So she actually has 3 different colors. So this is when her mom's activated. This is her dad. And then this is when both of them are not activated. So her white is whenever it's not showing either of
It's a
them.
little bit of what's it called? I don't know how to say the word.
She mixed epistasis.
Epistasis with a white.
She mixed.
A little bit. If
Yeah,
you
when it's not shown, it's
Yes.
epistasis. Okay, I get away from her. She's mad.
All right, we're gonna head back to the general area. And there we are. There we are.
We done?
We done?
No.
Oh, sorry.
So.
All right, sorry about that. So now we're moving on.
To the next.
Okay.
Well. Taste like disease.
Okay. Ooh, Tay-Sachs disease. Why did I get excited about that? My baby had Tay-Sachs disease.
Explain them.
Okay, so Tay-Sachs is like where it stops like a lot of neuro like pathways. Abby, no!
No.
I'm sorry,
The cat is.
the cat and the dog are kind of fighting right
Oh
now.
my word.
I'm sorry for
Oh
the
my God.
inconvenience.
Okay,
Do this
I'm
all
sure
the
that's friendly. That's
Friendly
Aw.
fire.
Brothers.
Okay.
Sisters.
Shut up.
both girl. Okay.
Okay. So Tay-Sachs disease is like a brain disorder in kids, and like most kids don't even get to like their 20s, even if it develops like later in their life. But if they're born, like if they express it as soon as they're born, they die by age 5 typically. So it's a.
Oh,
I'm sorry, we're looking at a diagram that's really cool. I'm so sorry. The labs are so cute.
So like they can't have like lipids, like they can't metabolize lipids. So that's kind of bad because we all need lipids to live.
No.
No,
It's
no.
not everything, obviously. Obviously, one of the 4 major components that are in our body is fine. If we don't have it, it's God.
But like only if they have both copies of the trait with Tay-Sachs do they have it.
Moving on to what I was thinking about the calico cat, epistasis.
Oh my gosh.
My
That's
pronunciation is a little off, but basically what this means is, I don't know, how do I describe it? Basically, if there's like 2 traits present, and they both are like activated, they kind of like shut each other off, and instead they turn off in a way.
That's kind of the perfect way to say it. It's like turning off a gene. If it was like a switch, that would be the analogy I would use.
Yeah.
It's turning it off and on. Which is a prime example of this was kind of the diagram me and Jose were like freaking out over. It'd be the labs. Technically, it's not that the dog is like a blonde or white color. It's just that their dark pigment was shut off, and that's why there are yellow labs.
It's not really co-dominance or dominance. They kind of just shut each
Just
other
yeah.
So the reason that I was kind of freaking out about the diagram is because 2 black labs have more chance of eating a like white lab rather than a brown lab, which is closer to like
the shade,
the
the
shade,
original
yes.
color.
which is
Kind of crazy.
Kind of crazy.
Genetics really went off away.
And then play trophy. Just how about that? Where is it? Play trophy.
And so we've been keeping typically one, like we've been stressing over the fact about how one gene will affect like the typical expression of a phenotype. And however though, you can have multiple of these phenotypic effects. Phenotypic effects, I'm sorry, I don't know how to say that. And this is called pleiotropy. And so we see that these alleles can often create symptoms and are typically the, I guess the factors that go into like many diseases such as cystic fibrosis and sickle cell disease. Within the lab that we were doing, my offspring actually had cystic fibrosis, and this disease caused lots of malfunctions within the it affected lots of parts of the body. Yeah.
In the lungs, where like mucus would build up, or in the liver.
Yeah,
Yes.
it's drastically shortens a person's lifespan, and it's really hard to breathe. Also, but I'd like to add on to that not all of these genetic mutations or like diseases that are caused by these genetic mutations are necessarily like super bad, because while sickle cell anemia is not like a favorable thing, it can be in certain areas of the world where like malaria is like predominant in like Africa or Latin America. It's you're kind of lucky if you have sickle cell malaria, sickle cell anemia, because you are resistant to malaria. You can't contract it, and that's kind of something that they would consider important.
And then like back to like the, I can't speak, but the trophy thing.
Pleiotropy.
Yes. Like it's like the different blood types, because I feel like we specified it enough. It's like the A, B, and O type of thing. Where like
O is the universal giver. A, B is the universal taker.
Whoa.
and then
Thank you.
so you always have to remember whenever you're doing like your pedigrees that you have like, if it's just A blood type, you gotta have that O, or if it's a double A, like always have those two, because it always two.
Pedigree. So we'll be jumping on to pedigree.
Wait, well, I have one more thing to say about the blood thing. You have to be very aware of the fact that, like, if you were to take in blood that isn't your own, your body does reject it, and that's something commonly like talked about in class, about how some mothers have different like blood types in their babies, and their like body rejects it and fights it off. That's how miscarriages occur. So I just like to call that attention. Okay, carrying on.
Carry on to pedigree, which is kind of a family tree, but instead of what it is actually showing is like traits being passed down from generation to generation, and seeing how they pass down, and seeing who or which people are carriers and which people aren't, kind of.
Yes.
And do you have an example for it?
In a pedigree, a circle is a female and a square is a male.
Yes. And in class, we've basically just been seeing the structure of these pedigrees. And so you'll typically see the very start of a pedigree with obviously the parents, as they are the root, I guess, of this family tree. And the reason that these alleles and genes go out to be and trickle down, that's why it's similar to like a family tree or so. And so whenever you're crafting these things, you're gonna need to see that there's like a connection between the two. And so here you have this line, and this basically just symbolizes the fact that this group right here is married. And so if they were to produce an offspring, the line would go down over here, let's say that they have, I don't know, let's say they have 3 kids. They have a girl, they got a boy, he's a square, and they got another girl. And so, let's say that both of the parents are normal. Let's say that a lowercase carries albinism, and let's say the capital is normal pigment. And so in this case, both of the parents are completely healthy, seeing that they have the dominant allele.
But how do you show that they're carriers?
Oh my word. All right, so you can typically show that a person's a carrier whenever it is shaded in and they have dominant recessive.
That's not carrier. That's if they. That's when they actually
Oh,
have
you
it.
silly goose, you.
A carrier would be if it is recessive and they're heterozygous and they carry it.
So like this person would carry it.
What?
So that you would do that.
So like for colorblindness,
What?
It's a sex-linked trait. So if
Oh,
your father
so they're
is
half.
colorblind, then
So this
it's more
person-
like- usually
This
the
person would have
girl,
all of it.
the
All of
daughter,
it, yeah.
will be a carrier.
Oh,
for
yes, of
colorblind.
course, then.
The same goes for like
Duh.
albinism.
So he's normal. This bad boy, he is half. That bad
That's a
girl,
girl.
I mean. And so then, let's say she marries someone. Let's say he's also a normal man. And the process kind of can repeat itself in that way. As you can see, they're obviously married. They got the little connection between them. And then they got another offspring. They're just going to have 2 daughters. Sounds good. 2 daddies. No, I don't like that. I don't like that. I don't like that at all. I don't like that. I do not like that. Alright, there they go. 2 daughters. And let's say, sadly, one's a carrier. Well, she has it. She has albinism. And this one's normal. So, Shadi has none filled in because she cannot be a carrier at all. She has a husband. Guess what? He's completely normal. Nothing. So, they got a daughter. She's safe. All of them, not carriers. All of them, no albinism. And that's all I gotta show you today for pedigrees.
Thank you, Aurelia.
Thank you for helping me with that.
No, thank
I'm not gonna cry, though.
Ah! Yeah, trip me on the way back.
We'll talk about the
You can't see me.
I'm sorry.
We're like a really important
Focus
part.
on that bad boy.
There we go.
There we go.
we'll just skip over to fetal testing.
This stuff like freaks me out.
Yes, there's only 2 types of fetal testing that can be done, which is amniocentesis and CVS. Which, you know, not like the pharmacy store, but
In case you're confused.
chronic villus sampling. And these 2 kind of distinct are very different from each other.
They take it from the placenta.
Yes, amniocentesis. Instead, they take fluid from the amniotic. They take amniotic fluid from the amniotic sac, but which is later during the pregnancy.
Yes.
But it is more accurate than the CVS testing because CVS testing, you could take some information that is from the mother and not
Yeah.
from the.
And potentially ruin the testing because
Yes.
it's inaccurate.
Yeah.
It also samples villus tissue, but it's earlier during the pregnancy, 8 to 10 weeks. But amniocentesis takes 14, 16 weeks, and basically that's about it. And these show you a picture of, well, not pictures. It
karyotype.
shows the karyotypes of the chromosomes. So, for example, if a baby would have an extra the 21st chromosome, then you would be able to know early in the pregnancy
That it has Down syndrome.
that it has Down syndrome.
This is particularly, I suppose, important when it comes to more pressing or severe illnesses. Like some mothers may use this information to decide whether or not they want to carry on with the pregnancy if the child is like fated to die at a very early age, which is very unfortunate and like sad. But it does help some couples better understand their situation.
It also just isn't for Down syndrome. It could be test for many other diseases or syndromes, if you will.
Associated with chromosomal mutations.
As in Tay-Sachs, Turner syndrome, Klinefelter, and many others,
Etc.
etc., etc. And
One thing that I found kind of crazy was like that Huntington's disease is a dominant trait. So like
dominant.
Yeah, that's
yeah,
scary,
it's
to be
weird.
honest.
It's like
It stays dormant for men.
For a long
For a
time.
long time,
Like
so-
well into their
Most people
adulthood.
most people who have it don't really know they have it until later in life.
until
when-
after like the fertilization or like pregnancy stage, like breeding stage in their life. So they
Yeah.
may have children, and then not realize it, and then they've already had children, so they pass it on inadvertently, which is kind of terrifying.
it's it's about your nervous system, and it basically degrades your nervous system, and it is fatal.
We have reached the end of our. Oh wait, are we not?
Adam has one thing to say.
Oh, my bad.
I was going to talk about polygenic inheritance, and that's when a single phenotypic character is affected by 2 or more genes.
Oh yeah, I forgot about that.
And that can be represented by this picture right here, where it's like one phenotype has like all of these colors, and that can affect like several things at once.
For example, what was it?
It can affect the
Like your-
phenotypic expression of.
Like your eye color or like hair color.
Exactly.
So
Is that where it affects like more than one thing?
Mm-hmm.
They were a good example of that for all different phenotypes of skin tones.
yes.
I'm red.
It's like me and Ryan, like the same.
No.
I'm
She's Pacific Islander.
Yeah, like I said, we're like the same.
same color.
Whoa. So yes, we have reached the end of this chapter.
On to the next. Possibly.
What?
No.
What?
Next.
Hey, next time.
Good
Next
job.
time.
Bye.